am going to be introducing Dr. Renaud La Joie. Thank you. He is an assistant professor of neurology at the University of California, San Francisco, where he provides leadership in Alzheimer's PET and MR imaging, fluid biomarkers, and heterogeneity in Alzheimer's disease. He trained in neuropsychology with Dr. Gail Chatelet in France, and then completed a postdoctoral fellowship with Dr. Bill Jagust at UC Berkeley. He is a star in his field, having earned several prestigious early career awards from Alzheimer's Association, two, in fact, from the Alzheimer's Association, also from Human Amyloid Imaging, UCSF, and the UCSF ADRC. Some of his recent work has been published in Brain, Alzheimer's and Dementia, and Neurology. He is the current recipient of an NIA K99 RO Award and Alzheimer's Association Research Award that focus on brain imaging and spread and propagation of tau in Alzheimer's disease. Again, thank you so much for agreeing to present your work. We're very glad to see you here and are looking forward to your talk.
Great. Thank you so much for the introduction. I'm really always very happy to give talks and talk about the work that not only I lead, but I work on with many other people at UCSF and beyond. So I will be focusing today on like multiple different types of study, but all looking at this issue, or this really key question of why is Alzheimer's disease presenting so differently in different patients. Here are my disclosures. As you mentioned, I get a support from the Alzheimer's Association and the NIA. I'm the co-lead of the neuroimaging core for our local ADRC. So all the data I'm gonna be presenting is from our ADRC. But before I actually talk about my science, I would like to talk a little bit about my trajectory, mainly because I've reached this point where I am no longer applying for training grants. And the good part of this is that I no longer have to explain my curriculum and try to justify my trajectory. So this is the plan I had originally when I graduated from high school in 2004. In France, you go to med school directly. It's like a combined grad program. I thought I was going to go to med school. Then I thought I was going to go to residency and be an adult, you know, quite fast after this. I come from this little town in Normandy called Cherbourg, and I was going to study in this other bigger city called Caen. I really the plan happened as as scheduled for 2 years, and then I had a major change, major event in my life. In applying for an MSTP program in France. It was, at the time, it was a national MD PhD program. It was not university specific, it was the whole national program. So after that point, I started to do rotations in research lab as I was also attending medical school, like the equivalent of pre-med in France. And I had to stop pause med school a little bit to have a full year of dedicated to research. And this is where the big first encounter, big meaningful meeting in my professional life happened when I met with Gail Chatelet, who was a researcher in Caen doing brain imaging and neuropsychology of Alzheimer's and aging. And really, she was a fantastic mentor. She was my first research mentor, really. And she convinced me to continue getting a PhD in neuropsychology. And that was really an amazing experience. During that PhD, I met Bill Jagust at AIC in 2011. It was not really exotic because it was my first AIC, but it was in Paris. So it was an international conference, the first I attended, but it was, a 2-hour train ride from home. And based on that, I knew Bill's work and I was really, really a big fan of his work, but I met him and I realized how great of a person he was. I think a lot of people on the call, and Laura can agree with this. So I managed to stop to get an additional break in my MSTP program to come and come to Berkeley, move to California and spend a year in his lab. It was really amazing. And after that, I had to go back to school in France. But actually, before that step, I want to mention that when I was at Berkeley, I then met with Gil Rabinovich at UCSF, who ended up being a very big collaborator, and this is, of course, very relevant for what's to come. So after Berkeley, I went back to med school in France, in this other place of France, and I really quickly understood that I really missed research, and I didn't get the point of why I would spend 3 more years of school and then a full residency and a fellowship to go back to research before I could go back to research, and I really missed research. And so I dropped out of med school and I moved back to the Bay Area to work with Gil at UCSF, and then transitioned to faculty later on. And the reason why I mentioned this is really because I was, it was a mix of having a nonlinear path and also being a non-US citizen, not being trained in the US. Was a lot of, try to summarize this on the BioSketch. It was a lot of work. I don't have to worry about this anymore, I think. But I just want to always mention this because I think a lot of trainees feel like they have to have a very linear path or a very straight story. And I think mine was not. And in the end, I think every little piece of it paid off and was really important. And of course, I want to mention these 3 mentors who have been really, truly amazing. And the choice of these pictures, there are many better, more professional pictures of them on the internet, but I think these reflect really how fun and amazing it's been to work with them. All right, and in that trajectory, I've worked on different topics, and I've expanded my research interest. You can see all these different themes in green on top, and then at the bottom, I've also really been very lucky enough to be in different labs where you could learn a lot of techniques and collaborate with a lot of people. So I won't be talking about all of this in this talk. I will, but what I want to say is that there's all these themes and all these techniques, but overall, there's a common denominator to all of this. And I think the key of my research is to use all these different tools, neuroimaging and other approaches for 2 main goals. The first one is to really try to better understand in vivo disease mechanism and why the disease is so heterogeneous across patients. And then the other one is really, and I care about this, I've always cared about this a lot, and I think it's getting more and more relevant today with a lot of controversies and potential use of new drugs in the real world, is to really help guide a better interpretation of biomarker data, in vivo biomarkers, and really do a better job at understanding what these biomarkers can tell us about the disease and what they also cannot tell us and what their limitations are. I think this is very important. There's still work to do. All right, so today I will mainly talk about this, about Alzheimer's disease, and especially so-called atypical presentations of Alzheimer's disease, mainly using PET imaging, neuroimaging, and neuropathology approaches. I will start by talking a little bit, defining a little bit what I mean by clinical heterogeneity, and then I will talk about both the role of tau and non-tau drivers of this clinical heterogeneity. So this concept might sound a little simple, but I think the way I like to describe is that we think of heterogeneity as a multi-dimensional concept. And I think there's different things that are related to each other, but we're going to try to study them one by one and in kind of like an orthogonal fashion. The first major difference between patients is the age of onset for their disease. We know that age is the main risk factor for disease. So disease incidence and prevalence increases with age, but there's still a lot of variability. And we like to look at this age of onset as a continuous variable. A lot of the literature is based on this threshold of 65-year-old to define to distinguish early and late onset Alzheimer's disease, but this is really a social construct in that it matches the age of retirement in most countries. But I hope that by the end of this talk, you're convinced that there's really value in looking at age as a continuous variable. And one thing I want to clarify is that there's around 5 or 10% of patients with sporadic Alzheimer's disease who develop Alzheimer's symptom before 65. But early onset Alzheimer's disease is often confused with autosomal dominant AD, And this is a figure from a review we wrote with Jenny Okuyama and her lab at UCSF, really emphasizing that most cases with early onset AD don't have a mutation on ABP or persillin. So most of these cases are sporadic early onset Alzheimer's. And this is what I will be presenting on today. I will not present on autosomal dominant forms of the disease. The other component is the clinical syndrome. And I think we all learned originally Alzheimer's disease as being like an amnestic, predominant, a slowly progressive dementia syndrome. But it's become really clear that this amnestic presentation was neither sensitive nor specific to capture Alzheimer's disease in everyone. And actually, the clinical criteria have changed. In '84, you need memory deficits to fulfill criteria for probable Alzheimer's disease. And it's no longer the case since 2011. And one of the reasons for this is that there's been a series of studies, a lot of studies showing that Alzheimer's disease could present as a non-memory predominant syndrome. And there are some of these syndromes that are listed here that are known to be highly predictive of underlying Alzheimer's pathology. The logopenic variant of primary progressive aphasia, you can think about it as a verbal presentation of Alzheimer's disease, posterior cortical atrophy as a more visual spatial predominant pattern. And then other variants are not always related to Alzheimer's disease, but can be due to underlying Alzheimer's pathology, like behavioral or dysexecutive variants of Alzheimer's disease, or even motor syndrome with critical basal syndrome. So if you plot this, the different cognitive impairment on this little plot here, one thing I want to really clarify is that these syndromes are listed here. They're kind of like very focal, very extreme cases of non-amnestic syndromes. And we study them because they are on this on the extreme ends of that spectrum. But this is really a spectrum, and most of the patients are going to fall somewhere in between, and they're going to have some deficits in one or more of these different domains. And the reason why we study these specific syndromes is just because they're like the extreme cases of heterogeneity. But of course, this is all spectrum. Another very important factor is the heterogeneity in clinical progression over time, and there's a lot of variability in that. This is a data not from us. This is a paper from a group in Amsterdam, I think, using the ADNI data. And here they plotted clinical progression, clinical decline with the CDR sum of boxes on the right, on the left, or MMSE on the right. And these are all patients from ADNI who all have an amyloid in their brain. We think they have some kind of, at least some levels of Alzheimer's pathology in their brain. They all start with a CDR of 0.5, and still you can see at the group level, you can run statistics and add these like thick black lines here to show the average decline. But there's a ton of variability, and no patient is this average line. So we need to understand what's driving this heterogeneity. Of course, I'm listing these three components as separately, but they're related. We're interested in studying their relationships and their association with demographic, genetic, and other factors. Like I said, they're not independent from each other. We know that some factors are going to have an impact on more than one. So, for example, we know that ApoE4, the main genetic risk factor for Alzheimer's disease and sporadic Alzheimer's disease, is associated with an earlier age of onset, but also a more memory predominant presentation. We also know, for example, that patients who develop their symptoms early on, so more early onset patients, tend to have a more severe clinical progression. So we're going to look at how all of this is connected, but also how it's connected with brain pathology. And for this, I will be presenting a series of studies using both imaging and neuropathology, with the big caveat that is bolded here, that I am not a neuropathologist. I have learned a lot from them, and it's been one of the most amazing things in being at the UCSF Memory and Aging Center. Collaborating with neuropathologists, I think, really increased my not only my understanding of the disease, but also it really informed my use of imaging. And I hope you will be convinced of this later. One thing before I present data, I want to clarify that in this talk, I will only present data from patients who have clinical symptoms. So I will only talk about the clinical stages of Alzheimer's disease, not the preclinical stage. That could be a different talk, but I think Bill Jagus gave a talk here a few months ago, and I'm sure he did this better than I would. As I mentioned, all the data I will be presenting is about sporadic Alzheimer's disease. We did not include anyone with autosomal dominant forms of the disease here. And I will show data from two cohorts, both from the UCSF ADRC. One thing that's important is that, so these two cohorts are absolutely non-overlapping, a neuroimaging cohort and an autopsy cohort, both of which have evidence for Alzheimer's neuropathology. So for the pathology sample, all patients have a pathological diagnosis of Alzheimer's disease. For the neuroimaging sample, all patients are amyloid positive. And the reason why it matters here is that the heterogeneity I will be describing, it has nothing to do with, patients who have a clinical syndrome of Alzheimer's disease, but kind of as a misdiagnosis in that they will have the syndrome due to a different kind of pathology. I'm really talking about patients who have both clinical impairment labeled as an Alzheimer's associated syndrome and underlying Alzheimer's pathology or biomarkers. And of course, a major caveat, the first part is not actually a caveat, but as I mentioned before, we are specifically interested in early onset AD and in these atypical non-amnestic syndromes. So our samples are enriched in these cases. And then the main caveat, and this is really common to most of the literature in our field, unfortunately, so far, is that the samples are non-representative. They're a vast majority of white, highly educated patients were recruited from tertiary clinics. So I think, and this is, the last few years, I've shown a lot of new initiatives to try to fix this problem. And I hope that next time I give a talk to this audience, maybe in a few years, we'll have a better data set to present in that regard. Right. So the first series of data I want to show is about neuroimaging data in patients with the different syndromes. So on the left, here you're looking at amyloid PET in 3 groups of patients. On the left, patient with an amnestic presentation of Alzheimer's disease. In the middle, patients with posterior cortical atrophy, so this more visual spatial predominant syndrome. And then on the right, a patient with the logopenic variant of primary progressive aphasia, so cases with the language form of Alzheimer's disease. And you can see that in all these patients, there's amyloid all over the brain, especially in the neocortex, and it really doesn't seem to be so different across syndrome. When you actually run a statistical comparison, the effects are very mild and they don't always make a lot of sense. They don't survive very stringent correction for statistical testing. And these results are weak and consistent with existing literature. The patterns are extremely different when you're looking at tau PET. And here we're looking at this tracer called flotalsip here, which is the tracer that's been used the most so far. It's the one that they use in ADNI as well. Here you can see that there's actually 2 things that are very important to notice. The first one is what's common across all these syndromes. And you can see that in all these 3 syndromes, you have a very temporal and parietal predominant pattern of pathology detected with PET, but then there's a lot of variations around that common area. Cases with posterior cortical atrophy have a lot more occipital and parietal, a very posterior predominant pattern of tau, and then the patient with logopenic variant PPA have this very strongly asymmetric left predominant pattern. And so, and of course, this is all very highly significant. You can see actually, it doesn't show so well on this rendering, but the patient with an amnestic syndrome have more medial temporal tau. So, it's this really clear, very easy to understand relationship where the tau PET pattern really mirrors the clinical syndromes. And that brings me to the neuropath data, because we're interested in what we're seeing in vivo, but we want to know how much it reflects what's going on in the brain and how consistent it is with the neuropathological information. And so this is data from UCSF as well, from Leah Greenberg's lab, who's a fantastic neuropathologist, and she really did develop with her research assistant at the time, Catherine Pedersen, a method to quantify tau tangle density in the brain. And the reason for this is that, this is something that I never really thought about as a neuroimaging and biomarker person until I really collaborate so strongly with neuropathologists. That when we read papers with neuropathology data and we want to know how much tau or how much of tau pathology patients have, we use this Bragg staging. That's really it's been used from probably 40 years at this point. And really, it reflects the stereotypical progression of tau from the medial temporal lobe to the cortex. And this is used for neuropathological diagnosis and many other studies. What I really did not understand so well until recently is that this is really not a good quantitative measure of how much tau there is in the brain. And this is just a plot showing the tau tangle density. This is data from the Rush Center, quantifying on the y-axis, you can see that the density of tangles in the entorhinal cortex. So this one of the first regions that get tau in the brain. And so by definition, everyone at Bragg stage, every patient that is labeled at Bragg stage 1 or above has tangles in the entorhinal cortex. But you can see that this amount of tangle in the entorhinal region, in the early region, keeps on increasing, at least at the group level, as tau propagates to other regions. But what's really important is that even in cases we have tau all over the brain, all the way up to the motor cortex in Bragg stage 6, there's still a lot of variability in how much tau patients have in their entorhinal cortex. And I think, this is really important because at the group level, again, there is a relationship between the patient Bragg staging and how much tau they have in their brain, in these early brain regions. But this is really not a good metric. And so there's really value in doing quantitative neuropathology. And I know that your ADRC is also doing this. And I will show some data from your group as well. But what's really important is that in this study, Leah and her lab really quantified a tau tangle density in different brain regions. You can see 4 different cortical regions, and then 2 regions in the medial temporal lobe, the CA1 and subiculum areas, and really showed that there was a lot of variability in how in where the tau was distributed. And all these patients have tau all over their brain, but there's still a lot of variability in where it is, and it really matches their clinical syndrome. You can see some syndromes that were presented here that were also present in my PET study, like the amnestic syndrome with more temporal, parietal, and hippocampal predominant presentations, and in patients with cortical basal syndrome with this motor predominant syndrome with a maximum burden of tau in the peripheral limbic area. So again, this data from pathology really mirrors our data with imaging, and that the relative distribution of tau burden matches the clinical syndrome. The other thing that was really obvious to us when we saw this is that the distribution of tau PET signal really mirrored what we could see with the MRI. And this is just in the same group of patient comparing brain atrophy across these groups using voxel-based morphometry. And this was really almost the same image. So patients who have more tau in their occipital cortex also have a lower brain volume. This is something that we see in the statistical analysis, but what was really so impressive that we could see this at the individual level. And when we looked at patients, we could see that tau really overlapped with regions that looked atrophied. The one thing was that in this group, we're looking at group averages. And what is important is to look at within these groups. And this is a study that looked at the same patients, but this time looking at asymmetry of amyloid and tau and MRI in these patients. I'm just going to show here like 3 examples of how much variability you can have in a group of patients. This is a tau PET image from a patient with very symmetrical tau pathology. You can see left, right is almost the same. This is a case with mild asymmetry. They all have like a typical Alzheimer's syndrome here. Mild asymmetry, you can see there's more signal on the right than on the left, but it's still pretty moderate here. And then you can have cases with a profound asymmetry like this. The problem is that when you're looking at group averages, again, you're washing out a lot of these asymmetry, because if half of the group is going to be left predominant and the other one right predominant, you will not see a difference at the group level. So we quantified this asymmetry in all the patients, and this is the data. We'll walk you through this. Again, we're looking at the same three groups of patients, our typical amnestic presentation on top, our logopenic variant PPA in the middle, and then the posterior cortical atrophy at the bottom. What's really very clear is that amyloid in general is very symmetrical. It doesn't show much asymmetry neither within group or between groups. And then the other point is that tau and atrophy are very asymmetric. They're very variable. And one thing that is very clear is that when you plot the asymmetry of tau and when you plot the asymmetry of MRI, it's really consistent across syndrome. You can see cases who have more tau on the right always have more atrophy on the right. And there's these other quadrants are really empty, showing that there's a systematic overlap between these two things. What is very important, though, is that there is a lot of meaningful results, like the logopenic variant PPA cases. The language presentation of AD is very left predominant in almost everyone. There's just one patient that's actually an outlier here. But this is this group is very asymmetric, but systematically on the left. The posterior cortical atrophy patients are interesting because they're actually all very asymmetric, but not systematically on the same side, which again goes back to the point of washing out a lot of effects when you're looking at a group average. The other thing that we were kind of surprised to find in these studies was that the asymmetry was extremely common also in typical presentation of AD. There's a lot of variability. It's not systematically on one side or the other, but there's a lot of variability. So this asymmetry is very is always found, at least in this study, almost always found in this non-amnestic presentation of AD, but it's not specific to them. And you can see a lot of asymmetry in typical cases as well. The other thing that we wanted to investigate was the relationship, and specifically the temporal relationship between tau and brain atrophy, because we really thought, and this is based on what we saw with single patients, but also based on what we think, how we think the disease works, that tau precedes brain atrophy. And so we had this patient who came back for a second visit a little above a year after the original scans. And we quantify brain atrophy with a specific pipeline. I won't go into details, but what I'm showing here is basically on the x-axis, you can see how much PET signal they had at baseline. So more, in blue, it's more the baseline level of amyloid. In green, their baseline level of tau burden at baseline. And then in yellow on the right, it's, excuse me, I'm plotting their baseline cortical thickness. So higher values mean a lower cortical volume at baseline. So basically looking at how abnormal their brain data was at baseline, and the y-axis is showing how much atrophy happened over the next year. Higher values meaning more severe atrophy. And you can see that, as expected, the more abnormalities you measure at baseline, then the more atrophy you observe over time. But this is really only significant for tau. And when you put all three biomarkers, baseline biomarkers, in a model, only baseline tau predicts how much atrophy is going to happen. But here, I'm only plotting global amount of brain atrophy, a general measure of brain atrophy. What we're interested in is this regional information. And this is really something that was, we're just putting statistics to quantify something that we could really see visually in single scans. And this is actually the same scan I'm showing everywhere in the slide. So showing, in the cortex where the amyloid was at baseline, here it masked for the cortex, where the tau was at baseline. And this image in the middle is quantifying how much atrophy happened over time, in like subsequent atrophy, with voxels in red, regions in red, showing where we observed the most atrophy over time. And we quantified the similarity between these maps with this correlation coefficient. You can see in this plot in green that the regions that started with a lot of tau PET signal actually were the regions that showed the most atrophy over time. So we're just putting statistics on this. This isn't a single patient, but we can repeat this statistics in every single patient with a lot of variability, but really just consistent effect of really mirroring image between baseline tau and future atrophy. And I just want to show this little video. These are 4 different patients selected to just show the range of variability. You can see in blue their baseline amyloid, In green, their baseline tau PET signal, and in pink, where atrophy was measured over time in that follow-up duration. And this is really, really impressive how much similarity you can see. The baseline tau really predicts where the brain is going to shrink in the near future. So this is really interesting to us, but we really care about what it means for patients and whether, the baseline tau patterns could help us predict not only where brain is going to atrophy, but also what is going to happen to the patient in terms of clinical changes. And this is a study in preparation from Mara Malpetti, who was a visiting scholar in our lab. She used tau PET data at baseline and then repeated cognitive assessments in around 80 patients with MCI and dementia. She modeled their clinical progression using latent growth curve models. So of course, this is not the real cognitive data. You'll know how cognitive data unfortunately does not look as linear and neat as this plot. This is the outcome of the model showing decrease over time in episodic memory scores. And when you look at the variability in these slopes over time, and you compare it to, and you correlate this with the baseline tau pattern, you really see that episodic memory decline was related to baseline tau PET signal, especially in the temporal lobe, in the here in more yellow voxels. And when you repeat this with different cognitive tests, it makes a lot of sense, and the patterns really mirror what we all know about brain behavior relationships. And this is, for example, the same analysis but repeated with decline in semantic memory over time, and patients relating the semantic memory decline to tau PET pattern, and really showing this mainly temporal predominant relationship. So this, tau PET patterns and as a single time point predict where the brain is going to atrophy in the near future, but also what cognitive decline is more likely to happen. The other component that I mentioned in terms of how variable patients are is their age of onset. And you can see here that when you plot how much amyloid patients have in our cohort, there's a lot of variability on this y-axis, but it doesn't seem to be related to how old patients are. So again, amyloid is not related to that variable. Things are very different with tau. Here we're looking at global tau burden in the whole cortex in our patients, and it's really strongly related to age, with younger patients having more tau. And this is something that we're curious to know if this was driven by these non-amnestic syndromes that tend to be younger. And it was actually not the case. Here you can see that the regression line was run separately in these three syndromes, and you can see that the lines are extremely parallel here. So within each syndrome, patients with a younger age of onset tended to have higher tau burden. This is again looking at global tau pathology measured with PET, and we're interested in whether it was like a global effect in the brain or if it was more local, more regional. And here in purple, you can see all these voxels, all these brain areas that show this negative relationship. So patients who are younger tend to have more tau in the frontal and the parietal cortex, basically in almost all the brain, except here in the medial temporal lobe. In the temporal lobe in general, we can see that the tau burden in the temporal lobe is in these patients is kind of independent from the age of onset. And I just want to mention a couple of studies from our fantastic, really incredible research assistant in the lab. She's looked at this tau relationship with age in two different ways. This is data from the LEADS cohort that, so it's like it's basically an equivalent of ADNI, but for early onset Alzheimer's disease. And our lab is the PET core, so we analyze all the PET data from this study. And so you can see by design, all the patients in this cohort are younger than 65 years old, but there's still some variability with our younger patients being, I think, 46. And even in that limited range of age of onset, we do see this relationship between younger age and higher tau burden. So this is really, again, 65 is not a magical number. We see this relationship between age and tau across all samples and age ranges. The other thing that I want to mention is that, and this is the only slide where I'm talking about blood biomarkers, but this is really an exciting development in the field. NITI looked at patients who had both tau PET and a measure of phospho tau in the blood using a specific assay here that looks at p-tau on residue 217. And she showed in this head-to-head comparison that this pattern of correlation between age and tau PET signal is also, we also find the same relationship with p-tau markers in the blood. So, and the reason why I want to emphasize this is that, it really tells us that these biomarkers, they show stronger signal in younger patients. So when we apply them, when in probably a few years from now, when we have access to these biomarkers, they will be extremely informative in younger patients. Between this is where we have the highest signal, and we have to understand how much we can learn in older patients when the signal is lower, and probably be very mindful of this. One point I want to emphasize with this relationship, again, I've been showing this negative relationship between age and tau quite a bit, but I want to emphasize that this is really based on cross-sectional data. So here, when people join the lab, they're always very confused by this result, because we're not saying that tau decreases over time. We're saying that younger patients, for a given amount of clinical impairment, younger patients have more tau. And there's this paper from the Mayo Clinic that showed that looked at longitudinal tau, so tau over time in the same patients, in different patients, and you can see that younger patients here between 50 and 65, they start the study with a lot of tau, and they keep increasing more. And older patients have less tau to start with, but they keep increasing. Their slope is a little lower, but all the groups increase over time. They just start from different levels. Here I will go back to this neuropathology assessment and the quantitative assessment of tau burden, just to show that the pathology data really replicated our PET study, with younger patients having more higher density of tau tangles in the brain, especially in these cortical areas. And when we looked at the medial temporal lobe tau, we did not see this relationship. So again, this is very consistent with our PET data. Tau burden in the cortex is really dependent on age of onset, but the temporal tau is more constant over groups. And this is a paper we just published, and at the same time, the UCSD ADRC published the exact same data with the same result in a very similar sample of patients, which was really beautiful, and we're so pleased to see this. So again, I'm just showing you a screenshot of paper from your group, so I won't go into details, but really it's with negative relationship between age and tau in cortical areas here, like the frontal cortex, but not in the hippocampus. So really, really constant data across centers. I mentioned this before, early onset of Alzheimer's disease is typically associated with more aggressive disease. So patients tend to decline faster and have higher rates of atrophy. And we were wondering if this is related to how much tau they have, and this relationship between younger age of onset and higher tau burden. This is data from just MMSE decline in our patients from the brain bank who all had Alzheimer's pathology autopsy. And you can see, even without statistics, that there's a very strong effect of age. With when you plot here, when you run a model and you try to estimate how much decline you can measure in patients in their 50s here at age 58, patients on average lose 20 points of MMSE over 5 years. And for the same time duration, a patient who's in their mid-70s would only lose 11 points. So we really see in this sample, we do see a major impact of the age of onset on the disease aggressiveness or disease course, really. So looking at the brain imaging data I showed before, we're interested in seeing if we see this the same effect with brain atrophy rates. And what we saw in this group of patients is that the atrophy rate over time, here as the y-axis, was related to age of onset, and our younger patients did show really more severe rates of atrophy over time. And we also know, we've shown before, that these younger patients have more tau, and we're able to show in a mediation analysis that the relationship between younger age of onset and greater atrophy rates was mediated by the fact that these younger patients have more tau at baseline. So there's really, all of this fits nicely together in that patients who have an earlier onset of disease have more tau to start with, and therefore more abrupt decline in brain volume and cognitive impairment. I will skip this one. So I think in the data I've shown before, I've shown a lot of like converging result between our imaging and our pathology samples, and really, one might wonder if, are we doing the same thing? Can we think of these two approaches as interchangeable and very similar? And I think the way I like to think about it is that there's really these are very complementary approaches. They're telling us different things, and they allow different projects, and they allow to answer and address different questions. And I want to show two quick studies that really show the power and the interest of each of these methods. This is data, we mainly contributed data to this big paper, but a paper led by Jake Vogel, who combined a lot of data from many different centers and many tau PET data from more than 1,500 individuals. And because PET is this like beautiful quantitative measure of pathology in all brain areas at the same time, you can run, you gather a lot of data in many patients in many brain regions, and you can run some really elaborate machine learning approaches to detect different patterns of tau. And I think this is something that is interesting. I've shown you the different tau patterns in patients that are classified as having different syndromes. In that paper, Jake did the opposite, and he used the tau PET data to derive data-driven subtypes of Alzheimer's pathology patterns. I won't go into details, but I think this kind of approach is really doing what's like using the best, leveraging all the information you can get from tau PET imaging in the numbers, both in terms of patients and brain sampling. Of course, the problem with neuroimaging is that we're gathering a single measure. We get this composite measure in the brain region. We measure some PET signal that is a mix of a lot of things that we're not exactly sure what the underlying pathology is. And there's more to the tau pathology is like a single number. And this is just a beautiful data from the Penn Center that looked at the same question we're interested in, looking at amnestic versus non-amnestic forms of Alzheimer's disease. And they use different antibodies to measure different types of tau pathology, different levels of tau tangle maturity over time. And with this antibody that measures both pre-tangle and mature tangles, you can see that so blue is amnestic AD, red is non-amnestic AD, and you can see that there's not a lot of there's a lot of variability across regions, but not really between groups of patients. And things are very different when you measure tau with a different antibody that captures more like end-stage tau pathology, these ghost tangles that stay in the tissue when the cell is dead. And then when you measure tau with this antibody, and when you're measuring these ghost tangles, you can see differences across groups. So again, really emphasizing that tau is not a single entity, is not a simple pathological feature. And depending on what you will measure, you will find different correlation with clinical features. And this is really important to be humble about what we're measuring with tau PET imaging, because we only get this one single composite measure. And this is, if you want to learn more about this whole idea of like tangle maturity, this is an amazing review by Melissa Murray and her group at Mayo Clinic that both describe the pathology very well for non-neuropathologists and also explain why it matters for biomarker development. And then the last part of my talk, I just want to talk really briefly about, I think I made a case for tau being related to the heterogeneity of patients' manifestations. But there's also all the non-tau factors. And I think there's a lot of papers that have been published showing that mixed pathology is the rule and not the exceptions in our patients, and that most patients with cognitive impairment actually have multiple pathology in the brain when they pass away. Most of this data comes from late onset Alzheimer's disease cohorts, and we're interested to know how it applies to our own UCSF ADRC cohort. And this is a study we did with Salvo Spina and another pathologist in the group, when in all our neuropathological cases with Alzheimer's disease, we measured the presence of other pathologies, so CAA, Lewy body, TDP-43, and other variables. And in this first plot, I am just showing the relationship between the age of onset in patients and how many pathologies they had in their brain on top of Alzheimer's pathology. You can see that there is a linear increase, like a very an association between the number of co-pathologies and age of onset, so that older patients have more non-Alzheimer's pathology than younger patients. But what's really, and this is kind of the relationship we expected, but what was kind of really surprising to us was to see that there were patients who developed the disease in their 40s or their 50s and still had up to 4 other pathologies in their brain, which really contradicting what I had heard before, that early onset AD is a pure Alzheimer's pathology. It seems to always be, like in most cases, patients also have other brain diseases. And this is a composite measure, but when we look into single pathology subtype, here I'm splitting patient in early and late onset AD just for the sake of display and to make it easier to understand. But you can see that for vascular lesion, for AGD, which is another type of tauopathy, for TDP-43, for hippocampal sclerosis, you really have this effect of age with older patients having more of these pathology, but this is not what we see in everyone for every pathology. For example, CAA, so amyloid beta deposit in blood vessels, and then Lewy body disease, so alpha-synuclein pathology. These two pathologies are both extremely frequent in our patient with AD, but they're also not related to age. And again, what was amazing with this is that the paper from your ADRC showed exactly the same results. So you can see Lewy body pathology was not related to age. It was frequent, but it was not related to age of onset in your autopsy cohort. In contrast to TDP-43 or arterial sclerosis that increase in prevalence with older age of onset. So this data and our data really show the same of some pathologies being associated with older age of onset, while others are just often present with AD pathology but unrelated to age. One factor that we're very interested in is to know how big of a deal that issue of copathology was in patients, and does that impact patients' clinical presentation? And this is again showing the MMC data in our cohort, but splitting patients between late and early onset, and then showing how many copathologies they had in the brain on top of Alzheimer's. And when we run this into a model, what we could see is that you can have the full line are showing MMSE decline over time in patients with early onset. The dashed line are patient with late onset. Sorry. So you can see that the decline, as we mentioned before, is steeper, is more severe, is faster in patient with early onset. But on top of this, patients who had more copathologies at autopsy, so red lines, had lower MMSE than patient with fewer copathologies, and that was observed across both groups in both early and late onset. So, we're like probably a little underpowered in this study to really detect a strong effect of the presence of copathologies on the rate of decline, but it seemed to matter at least to explain some of the level of cognitive decline we can measure in patients. One point I would like to end with, in terms of considering the copathology issue in vivo cohorts with biomarkers, is that and I think this is to justify my training in neuropsychology. I will be using a cognitive metaphor to explain what I think is happening when we're looking at biomarkers in vivo. So thinking about hemi-neglect and how patients with hemi-neglect are not able to really represent the reality was presented to them and can only see part of it, or perceive part of it. I think this is what's going on with the biomarker in the biomarker field. So we have like a multitude of different pathologies that might be happening in the brain. We don't know if they're happening or not, but we know that all these things can happen. But the problem is that the biomarkers that we have access to, and the biomarkers that are currently validated and that we're pretty confident with, they can measure tau, they can measure amyloid, they can also measure cerebrovascular lesions. And I didn't talk about this so much, but this is something we can measure with MRI, with other techniques. And we can get, it's not a perfect copy, but we can get a sense of of whether these things are happening or not. The problem is that there are all these other pathologies that might be in the brain, that might be there, but maybe they're not. And we can't tell directly because we don't have good direct biomarkers to measure them in vivo. So we're talking about TDP-43, we're talking about non-AD tauopathies, and also Lewy body disease, although I'm hopeful that this little pink box might move to the right, maybe in the near future with the development of new assays to measure alpha-synuclein. We'll see. That would be really interesting to be able to measure this in vivo. But the issue with this problem of having biomarkers for some, but not all the pathologies that, of course, if we have a patient with cognitive impairment and they're negative for amyloid and tau, we will therefore assume that it's because something else is happening in their brain. The problem is that even when the AD biomarkers are positive, we don't really have a good way of knowing if Alzheimer's pathology is the only or even the primary neuropathology that's causing the trouble. And I think this is really, really a big issue we have in the field, especially if anti-Alzheimer's pathology therapies are used. Do we know? Can we know how much of the clinical syndrome is due to underlying Alzheimer's pathology? So I will skip that part. I will just go to my conclusion. Well, maybe just to show briefly some APOE4 data. We showed that in our patient who had APOE4 had more tau in their temporal lobe, in their medial temporal lobe, and that was really robust. That was a very focal effect. APOE4, this main risk factor for AD, was really associated with tau, but only in the medial temporal lobe, and that was really robust. And that was also very consistent with different groups, like findings from different groups. This is a group from Montreal looking at ADNI and their local cohort. You can see the very focal effect of E4 on tau in the medial temporal lobe. And this is data from Boston showing again, like, while age and amyloid have major effect on tau burden, APOE4 has this very, very, very focal effect on tau burden in the medial temporal lobe. So really, really reproducible finding. I will skip this for the sake of time and just we'll jump to my summary slides. So I think we've shown that in multiple cohorts and consistent with other groups, the clinical variability of Alzheimer's presentation was associated with tau and not amyloid differences. Again, from PET, from pathology, from our group, from your group, from many other groups. What we saw, and I think this is really important to think about, is that both older age and APOE4, we need to think very, very thoroughly about them, because we know they're both the main risk factors for Alzheimer's presentation, but they're not simple risk factors. They also impact the way the disease presents and the type of symptom patients present, because they impact the way tau pathology is distributed in the brain. Both factors are associated with a higher with a more predominant medial temporal lobe predominant tau burden. So more tau in the medial temporal lobe compared to the cortex. So it's consistent with this amnestic presentation. But what's interesting is that both of these factors seem to increase the tau, like the MTL tau versus cortex tau ratio through a mechanism that does not involve amyloid beta. We need to figure out why, why tau patterns vary so much across cases, whether it's genetic underpinnings. We don't really know much about this right now. There's another hypothesis that I think is really interesting, is the idea that the tau pathology happens to different brains, and that the pre-existing brain architecture actually drives a lot of how the pathology is going to distribute in the brain. And there's data suggesting that patients with this atypical syndromes of AD have a higher prevalence of developmental differences. So patients with logopenic variant PPA, they actually had higher rates of dyslexia, developmental dyslexia. And so with the idea that it's not saying that dyslexia is a risk factor for Alzheimer's disease, it's saying that dyslexia might drive, or some of the brain changes that are associated with dyslexia might drive the tau pathology to deposit in different brain areas if Alzheimer's pathology happens. And I think this is something that And, it's still very hypothetical for now, but I think this is really worth investigating. Again, younger patients have more tau, especially in the cortex. We don't know if maybe it's because the pathology is slightly different, if there are some biochemical differences. And like I mentioned, maybe it's just that it's the same pathology that is happening to different brains. The higher rate of the higher levels of tau in young onset might be due to the fact that they have a little less co-pathology. So maybe they need more tau to get to a clinical level for a clinical syndrome to appear. And of course, there's this issue of, how hard it is right now in living patients to understand and to really interpret how much of the clinical impairment is due to Alzheimer's pathology versus all the other things that we are not able to measure. And so I will jump directly from that slide to my thank you slide, just to highlight all the people who did all the work. Here we go. I'm sorry. Here we go. Just wanted to thank everyone at UCSD, Gil Rabinovich lab, Jacob's lab at Berkeley, and then all the current and past mentees I've had really the chance to work with, and all my mentors, collaborators, or maybe even some of the former mentees that are now collaborators or maybe even mentors now. So thanks a lot, and thanks for your attention.